Good morning, students. Today we are having lecture number 7. Today we are starting to study special microbiology. Maybe you remember that microbiology can be divided into general and special microbiology. Previous lectures from number 1 up to number 6 were about general microbiology, about general structure of microorganisms, their metabolism, their different habitations where they live. Now we are going to start to study microorganisms that cause infections in human. Firstly, I want to remind you that next week all groups will start to have colloquium. Next week and after next week. In all groups, colloquium will be orally. Today we are having lecture number 7, part 1. Next time I will explain part 2 because this lecture is so large and it is impossible to explain you 8 species of bacteria in one lecture. So today we are going to talk about 4 species of bacteria. Firstly, we are going to talk about clinical microbiology. Clinical microbiology, what is it? We should say about it. Then we should say what are causes of hospital infections. Clinical microbiology studies the interaction between human organism and microorganisms in norm and pathology. Then, in the dynamics of the inflammatory processes, its diagnosis and treatment of infections caused by microorganisms, especially hospital infections. Once more, let's look at this definition. Clinical microbiology studies interaction between our organism and microorganisms in normal conditions, in normal when human is healthy, when there are no any diseases, and interaction of our organism and microorganisms in pathology, in diseases. Then clinical microbiology studies interaction of human organism and microorganisms when patient has inflammatory processes and how it develops, and then it studies diagnosis and treatment of these infections. Especially clinical microbiology studies interaction of pathogens that cause hospital infections. What are hospital infections? Some doctors call them nosocomial infection. Nosocomial infection, it's the onset of infection in the hospital. What does it mean? Let's imagine that our patient is delivered to a hospital in surgical department, for example, for operation, and there, after operation, this patient is infected by any microorganisms, any bacteria or viruses in hospital. Or another patient with, for example, high blood pressure. Let's imagine that the patient with high blood pressure has treatment at hospital, and there he gets infected by any microorganisms. It's called hospital infection because bacteria always present at hospitals. Bacteria and viruses. What are causes of hospital infections? What are causes? Why do hospital infections happen? The first group of causes are microorganisms causes. Hospital infections occur because there are antibiotic-resistant microorganisms. These microorganisms are present and they are survived on the surface of some medical devices, some medical equipment, for example, artificial ventilators, in catheters. Some medical equipment is extremely difficult for sterilization and for disinfection. It is impossible to kill bacteria in medical equipment by some substances like alcohol or chlorides. Another cause of hospital infections here are high virulent microorganisms. Almost all bacteria that cause hospital infections are very high virulent. They have a lot of virulence factors. What are virulence factors? I hope you remember from lecture number 4. Virulence factors includes adherence, toxin production, exotoxin or endotoxin. Aggressiveness, it is production of aggressive enzymes like hemolysin, hyaluronidase, coagulase. So, such bacteria that cause hospital infections are characterized by large amount of virulent factors. They are very high virulent. Another group of causes of hospital infections are human causes. It means that in these cases, onset of hospital infections depends on human. Who can it be at hospitals? The first one, these are risk group people. Risk group people are bacterial carriers among patients and doctors. Bacterial carriers or any sick patient, for example. Who are bacterial carriers? In lecture number 4, I also explained it. Bacterial carriers are patients without any clinical manifestations, but they have bacteria in organism and they able to infect any another human. For example, of bacterial carriers in Staphylococcus aureus. Staphylococcus aureus live in nasopharynx, and it's very dangerous when doctors or nurses or any other medical staff have Staphylococcus aureus in nasopharynx because they can infect other patients. A lot of patients at hospitals have low or weak immunity. Patients after operation, after surgical incisions, after radiation in cancer, for example, they have weak immunity. That's why they're very sensitive to bacteria. Bacteria cause hospital infections in patients. Another cause of this group of causes are immune deficiency human. Humans that I have said, these are patients with low immunity. Which microorganisms cause hospital infections? Any group of microorganisms or agents are able to cause hospital infections. These are bacteria, for example, Escherichia coli, Staphylococcus, Pseudomonas, Klebsiella, Streptococcus. These are fungi like Candida and Aspergillus. These are viruses like herpes viruses and adenoviruses. And protozoa microorganisms like Pneumocysta and Toxoplasma. We also should say that hospital infections are often caused by opportunistic bacteria. I also said about it in lecture number 4. What are these bacteria? Opportunistic bacteria, these are normal microflora bacteria causing diseases in special conditions, for example, in weakened immunity or in destruction of some barriers, of some mechanical defense barriers like skin and mucosal membrane, when this bacteria can enter other organs. So in most cases, hospital infection is caused by opportunistic bacteria. These are bacteria that are normal microflora, for example, in intestine or in nasopharynx or on the skin, and they cause disease in patients with low immunity, like E. coli, Staphylococcus, and Streptococcus. What are features of opportunistic infections? We should say that opportunistic infections are distinguished from other infections in patients. For example, opportunistic infections are distinguished from infections caused by other bacteria like Mycobacterium tuberculosis or by Salmonella typhi, or maybe Vibrio cholera. So opportunistic infections are distinguished from other ones. Once more, opportunistic infections are infections that are caused by bacteria of normal microflora in special conditions, in weak immunity, for example. At first, the source of infection, of opportunistic infection, are patients, bacterial carriers, and objects of the environment. Which objects? If it happens at hospital, this is medical equipment, maybe it can be shower, toilet, so any other objects. The another one, the causative agents of opportunistic infection may affect different organs and systems through any route. For example, E. coli or Staphylococcus. Staphylococcus cause more than 100 different infections. Staphylococcus cause infection of soft tissue and skin infections. This pathogen also causes infection of cardiovascular system, infections of nervous system, infections of musculoskeletal system, infection of digestive system, a lot of different infections. Or E. coli. E. coli can cause intestinal infection. In newborn, it causes meningitis. Then it can cause pneumonia sometimes, not very rare, but sometimes it can be. Then it can cause infections of urogenital system. So in any organs and system. Opportunistic infections appear in immunocompromised patients. Immunocompromised patients are patients with low immunity. It can be patients after radiation, surgical patients, it can be patients with HIV, it's also old patients, for example, who are elder than 50 or 60 years. They are sensitive. They are susceptible to opportunistic infections. Opportunistic infections are very often endogenous infections. Let's come back in our mind to lecture number 4. There are endogenous and exogenous infections. Endogenous infections are caused by microorganisms of normal microflora. We have said about it. The another one, opportunistic infections are mixed, generalized, and chronic infections. All these words must be known for you from lecture number 4 too. Mixed infection means that such infections are caused by some species of microorganisms, more than one species. Generalized means that opportunistic infection spread in human organism via blood, lymph, or nervous. And chronic means that these are long-term infection, long-term that lasts during some months or even some years. Then, opportunistic infections are very badly treated because most pathogens are resistant to antibiotics. Opportunistic infections are widely spread in hospitals, in all hospitals, and they are very resistant to disinfectants. Disinfectants are substances that kill bacteria. For example, alcohol or chlorides, they are used for washing of some medical equipment. And another one opportunistic infection are often connected with medical manipulation because they mostly occur after or during surgical operation, after injection of catheters, even after when a doctor or nurse collect blood from the patient in any medical manipulation, if medical equipment is contaminated with microorganisms or if medical staff are bacterial carriers or sick. Then, so we have said about hospital infections. These were general questions of hospital infections, and now we are starting to study microorganisms that cause infections in human. Look at this scheme. This scheme is used for description of any microorganism, of any pathogen. If you take any textbook in Russia, in India, in Egypt, in the United States of America, in any European country, if you take microbiology textbook, you can find out that any bacteria or virus is described by using of this scheme. What does it mean? It means that we must describe biological features for any pathogen. What are biological features? Firstly, we must know what is Latin name of family, genera, and species of this bacteria. Then we should say what about morphology of this pathogen. What is shape, gram staining, is it motile or non-motile, it has capsule or doesn't have. Then we talk about cultural properties. These are nutrient media that are used for its cultivation. And what about colonies? Which colonies are produced on the surface of the medium? Then we are talking about fermentative properties that include proteolytic, peptolytic, and carbohydrate splitting enzymes. Why do we talk about them? You remember that all they are used in identification of bacteria. Then we talk about antigens and virulence factors. Virulence factors includes toxins. We must know which toxins are produced, aggressive enzymes, for example. So we study biological features because, firstly, they are used in identification of pathogen. You must know them. And then, for example, we study virulence factors here in biological features because most clinical manifestations of disease that we see in patient are caused by virulence factors in bacteria. If we see diarrhea in patients with cholera or typhoid fever, we know that it is caused by toxin. Or if we see dehydration in patient with cholera, it is also caused by toxin chloragen in cholera. So we must know because we can fight with it. We can neutralize toxin of bacteria. The another point or the another characteristic that describes any pathogen is epidemiology and pathogenesis of diseases caused by this pathogen. In this question, we should say about diseases that are caused by this pathogen. You must remember which diseases are caused by any bacteria. Then we should know what are sources of infection. What are sources? These are human, animals, or environment. Because you are future doctors, you must know how to prevent diseases, how to prevent spreading of diseases. Then we should say about transmission ways. How is it transmitted from sick human or animals? And we should say about pathogenesis. Pathogenesis means includes features of diseases, its clinical manifestations, changes in affected organs and tissues, and clinical symptoms. In pathogenesis, we also talk about development of disease, how it develops in human organism. And then microbiological diagnosis, treatment, and prophylaxis. In this question, we will talk about specimen, which specimen must be collected for diagnosis. So which material? It is blood, or maybe feces, or urine. Then we must say which methods are used for diagnosis. Which methods are used for diagnosis. And then we should say about treatment, which medicines, for example, antibiotics are used for treatment, and about prophylaxis and prevention. Are there or there are no any vaccines and serum for prophylaxis or prevention? It's vaccination and immunization. So any pathogen will be described by this scheme. Let's start from the first pathogen. Today we are talking about hospital infections, that occur in hospitals. So we will talk about pathogens that cause hospital infections in patients. The first pathogen that we discuss is Klebsiella. And the first question that we discuss here is biological properties of Bacterial Klebsiella. I want to come back to this scheme. Look at this scheme, please. So here we are going to discuss all these 6 sub-questions. In biological properties, we are going to discuss this one. Firstly, we should say about its nomenclature, about its family, genus, and species. Family is Enterobacteriaceae. Genus is Klebsiella. Species are Klebsiella pneumonia, Klebsiella ozena, and Klebsiella rhinoscleromatis. Firstly, look please once more, pay your attention at name of family. Entero means intestine. So Klebsiella is a member of normal microflora of human intestine. So then you will study other bacteria and they are belong to this family. Enterobacteriaceae means they live normally in intestine. Name of genus Klebsiella. Names of genus very often were given in honor of scientists who discovered this bacteria. So this bacteria was discovered by Dr. Klebs. And species. Name of species also very often have interesting names because they were named because of diseases caused by them. Klebsiella pneumonia cause pneumonia, Klebsiella ozena cause ozena disease, and Klebsiella rhinoscleromatis cause rhinoscleroma. Pay your attention, if we write a full name in Latin, full name of genus, then we should write short name of genus in description of species. We write just the first letter of genus. This rule is used by a microbiologist. We shouldn't write the full name of bacteria once more if it was written once fully. Morphology of Klebsiella. Look at this diagram. It is a smear gram staining. These are gram-negative straight rods. They are non-motile. They are capsulated, so we can use special staining for it. Varus staining for detection of capsule. And they are non-spore forming, don't produce spores. What about cultural properties? They are facultative anaerobes. I hope you remember from lecture number 2 that facultative anaerobes can live in both presence or absence of oxygen. This pathogen can grow on simple media like meat-peptide agar and special media, for example endomedium and McConkie agar. Remember please that endomedium and McConkie agar are differential media for Enterobacteriaceae. We will study other pathogens from this family, so you can memorize, you can remember that other pathogens from family Enterobacteriaceae are also cultivated on endomedium and McConkie agar. These bacteria form smooth, colorless colonies and large red colonies on endomedium, for example, like here, red or pink. The another, fermentative, sometimes it's called biochemical properties. This pathogen ferment lactose and hydrolyze urea. I remind you that biochemical or fermentative properties like carbohydrate splitting enzymes, proteolytic enzymes, and peptidylitic enzymes are used in identification of pathogen. Antigenic structure. You studied lecture number 6, so you remember that bacteria can have O antigen, H antigen, K antigen. What about Klebsiella? It has K or capsular antigen, that is polysaccharide. It is thermostable, not destroyed with high temperature. And another antigen is O antigen. It's the lipopolysaccharide of cell wall. It is present in cell wall, thermolabile. If you remember morphology of bacteria, you can memorize antigenic structure. You remember that this bacteria have a cell wall, so it has O antigen. And you remember, let's come back. You remember that it has capsule, so it has K antigen. What about virulence factors? Klebsiella have strong adhesive properties. It means that their ability to adhere, to attach to mucosal members is very strong, is very high, because they have pili and capsule that are responsible for adherence. And adherence is the first step of any infection process. Any infection process starts from adherence of pathogen on mucosal membrane. K and O antigens that are present in Klebsiella protects against phagocytosis. What is phagocytosis? Maybe you know from biology. Phagocytosis is a process of killing of pathogen by phagocytes. When phagocytes, these are leukocytes, red blood cells. leukocytes, neutrophils, for example, they engulf pathogen and digest this pathogen. But this Klebsiella has K and O antigen that make bacteria resistant to phagocytosis. So bacteria is not killed. This immune mechanism is not active, and this pathogen have endotoxin. Once more, let's come back to lecture number 4. Bacteria produce two types of toxins, endotoxin and exotoxin. Gram-negative bacteria in most cases produce endotoxin. So, you should memorize what is gram staining of this pathogen. That's why you can say which toxin is produced. Endotoxin causes toxic shock in patients when there is a large amount of endotoxin in the bloodstream, and it can cause death during short time. So we have just discussed biological properties of Klebsiella. Another question is epidemiology and pathogenesis of diseases caused by Klebsiella. We have said about biological properties. Now about epidemiology and pathogenesis. Firstly, you should know that Klebsiella is a member of normal intestinal microflora. It lives in intestine in all humans. The source of infection is sick man and bacterial carrier. When Klebsiella enter other organs and tissues, it causes infections. So these infections are transmitted from sick man and bacterial carrier. How are these infections transmitted? They are transmitted by direct contact with environmentally contaminated surfaces, by fecal oral or airborne transmission via secretions that contain bacteria, from nasopharynx, in speaking, in coughing. Which diseases are caused by Klebsiella? We talk about it in pathogenesis. Firstly, it's pneumonia, because Klebsiella pneumonia causes pneumonia. Pneumonia, I hope you know what is it. Pneumonia is inflammation of lungs. Symptoms of such pneumonia include fevers, chills, and leukocytosis with red, current, jelly-like sputum. The another disease are bacteremia and sepsis. You must be ready that I will ask you what is bacteremia and sepsis. You may find this definition in lectures, in other lectures. Lecture number 4, you studied it. Bacteremia is a circulation of bacteria in the bloodstream, and sepsis multiplication of bacteria in the bloodstream. Bacteremia and sepsis occur especially in newborns. At hospitals, who are infected via some medical manipulations. Then, Klebsiella causes urinary tract infections, especially in patients with catheters, with urinary catheters. Some patients who are not able to discharge urine after operations, these patients have catheters, and when doctor or other medical staff inject this catheter, he can contaminate urinary tract of the patient with Klebsiella from the hands, for example. It's a direct contact transmission. Then wound infections. And Klebsiella also causes two specific diseases that are caused by just by this pathogen is ozena. You remember Latin name Klebsiella ozena, and rhinoscleroma. Ozena is a chronic atrophic rhinitis. Rhinitis is inflammation of nasopharynx. Rhinitis with necrosis of nasal mucosa and mucopurulent nasal discharge. It's rhinitis with necrosis of nasal mucosa and discharge of mucus from nose. Rhinoscleroma is a chronic inflammatory disease of nasopharynx with a purulent nasal discharge. Formation of nodules that may lead to respiratory obstruction. Because nodules are formed in nose, they stop transmission of air, and it is impossible to breathe for the patient. Both ozena and rhinoscleroma are chronic diseases. They occur in patients. They last during several years, during 5 or more years, and both diseases leads to inability to breathe. Microbiological diagnosis, treatment, and prevention of diseases caused by Klebsiella. Firstly, before to make identification of pathogen, we should collect specimen from the patient. Specimen is material collected from the patient. Remember, please. Which specimen is collected? Once I have said about it, once more. The choice of specimen depends on infection. Depends on which infection is caused by this pathogen. Let's come back to cause disease. If you don't know which diseases are caused by this pathogen, you are not able to collect specimen from the patient, because how can you know which specimen must be collected if you don't know in which organ bacteria is present? For example, if pathogen causes pneumonia, if our patient has pneumonia, what can we collect from the patient? How do you think? What can we collect? In pneumonia, we can collect sputum, because bacteria are present in lungs. Then, if our patient has bacteremia or sepsis, we can collect blood, because it's presence of bacteria in the blood. In urinary tract infections, how do you think? What can we collect? In UTI, urinary tract infections, it's urine. In wound infections, we can collect discharge from wound or pus from wound. In ozena and rhinoscleroma, we should collect, how do you think? We know that ozena is infection that occur in nose, so it's nasal discharge from nose, and rhinoscleroma is also infection of nasopharynx, so we should collect discharge from there. So specimen includes blood, serum, urine, pleural fluid, it's from lungs or sputum, wound discharge, nasopharyngeal discharge. We collect serum because we use serological test for diagnosis. Serum is taken in almost all infections. Which methods are used for diagnosis? The first method that is used is microscopic. Microscopic method includes direct microscopy with gram stain smear and Barry's method for detection of capsule. You remember this procedure. We should take a smear, a slide, then prepare smear, stain it by gram staining, and observe in microscope. Firstly, we should understand it's gram positive or gram negative. And then, bacteriological method. It's also sometimes called cultural method. We have said about its in lecture number 2, but I want to remind you how is this method used. Bacteriological method, remember please that bacteriological method is also similar, is almost similar for all bacteria. That's why you should remember it just once, and then it will be same for other bacteria. On the first day, we make inoculation of specimen on endomedium, put it in thermostat for 18 up to 24 hours at optimal temperature 37 degrees Celsius. And second day, we should prepare smears, inoculate material from colony of slant agar to isolate pure culture. We should put slant agar in thermostat for 18 up to 24 hours at optimal temperature 37 degrees Celsius. And on the third day we should make examination of morphological, its shape, gram staining cultural, how bacteria grows on media, its biochemical properties or fermentative properties for identification of bacteria. This scheme of bacteriological method is the same for every pathogen, for every bacteria, but they are cultivated in different medium. That's why bacteria are distinguished just in media that are used. Some bacteria grow longer. I will tell about it. Then another method are serological method, serological tests that detect antibodies. Which serological tests are used? It's immunoelectrophoresis and agglutination test. Mechanism of these tests and how they are used will be studied in immunology next semester. Now just memorize it. And molecular method, that is PCR, polymerase chain reaction, is also used for detection of pathogen. What about prophylaxis, about prevention? There are no any vaccines for prevention of diseases caused by Klebsiella. Immunity is absent after diseases. It means that patient can be infected a lot of times during all the life. But I said that most patients have chronic infections. What about treatment? Klebsiella is a pathogen that causes hospital infections in patients, wound infections, pneumonia, bacteremia, and sepsis. It means that this pathogen is resistant to antibiotics. That's why we should use disc diffusion test to detect sensitivity of bacteria. You remember we have talked about this test, disc diffusion test. Which antibiotics can be used for treatment of the patients? These are imipenem, M, amoxiclav, and cephalosporins. Klebsiella are fully resistant to ampicillin and amoxicillin. So these antibiotics, ampicillin and amoxicillin, are not used. Another pathogen that we discussed today is Proteus. Proteus, the name of pathogen, was given in honor of Greek god. There were a lot of gods in Greek history, so it was given in honor of this god. Family is the same, Enterobacteriaceae. Genus is Proteus, and there are two main species that cause infections in human: Proteus vulgaris and Proteus mirabilis. Morphology of this bacteria. Proteus are gram-negative, rod-shaped, motile bacteria. They are peritricious. They have flagella that cover all the surface of bacterial cell. Non-sporing, non-capsulated. Klebsiella. If we compare, Klebsiella was non-motile and capsulated. If we compare these pathogens. In this diagram, you can see smear of gram staining of Proteus. Cultural properties. This pathogen is facultative anaerobe. It's the same to Klebsiella because it can live in both with oxygen or without any oxygen. This pathogen can grow on simple media like MPA, and it also can grow on special media like McCone-Keger. And here we can see that Proteus produce yellow colonies on its surface. On blood agar, we can see swarming on all the surface of agar. What is swarming? Look here, let's compare colonies here and here. We can see spherical colonies on McCone-Keger, but on blood agar we can't see any spherical colony. Bacteria produce growth that is similar to some circles. Why does it occur? Because on blood agar bacteria are very motile, and they move very fast on the surface of blood agar. When they move, they spread colonies and their growth over this surface. This specific property is present in Proteus mirabilis. Mirabilis is translated as amazing from Latin language. That's why it's because of specific growth. And another specific property of Proteus it's fishy smell. If we open petri dish, we can smell it, and these are two specific properties for Proteus. It's growth, swarming, when it produces circles on the surface, and fishy smell. Biochemical properties. It has positive urease test. It means that Proteus produce urease to hydrolyze urea. It produces hydrogen sulfide. That's why we can smell fish, like fishy smell. It ferments glucose and maltose and don't ferment lactose. These properties are used in identification of pathogen. It has antigen, it's somatic lipopolysaccharide of cell wall because it has cell wall, and it has H antigen because it's motile, it has flagella. What about virulence factors? This pathogen has pili or fimbria that are used for adherence by bacteria, for adherence to mucosal membranes. This pathogen produces protease. These enzymes cleave secretory IgA. IgA is antibody that take part in destruction of Proteus in human organism, but protease destroy antibodies, so antibodies are not able to fight with infection. This pathogen produce urease. It's enzyme that provides a suitable or good environment for surviving of bacteria. This pathogen produces urease that splits urea into ammonium. So these conditions are very good for bacteria. It also produces hemolysin that damages red blood cells. Mortality is also a virulence factor because it initiates the colonization of the urinary tract. These bacteria are very motile. They have flagella that cover over the surface of bacterial cell. That's why bacteria very motile. It can colonize the mucosal membrane of urinary tract very fast. And this pathogen produces two types of toxins. The first one is endotoxin, because this pathogen is gram-negative, and enterotoxin. Enterotoxin is a type of exotoxin. It causes diarrhea. Then we should say about epidemiology and pathogenesis of diseases caused by proteus. The sources of infection are sick men and bacterial carriers. Infections are transmitted by contact transmission, foodborne, and airborne transmission. Transmission ways of proteus are similar to Klebsiella. Proteus are also normal microflora bacteria that produce infections in humans only when the bacteria leave the intestinal tract. When bacteria leave intestinal tract and enter other organs and tissues, or it can cause infections in patients with weak immunity. What are these diseases? The first group of diseases are urinary tract infections. These infections occur in patients when they are injected via catheters, urinary catheters, for after some operations. When proteus enters in urinary tract, it produces urease. I have said about this enzyme, urease. Urease splits urea into ammonia that has a pH about 9, and it leads to production of renal stones in bladder or in kidneys. The another infection is caused by proteus, or is bacteremia, that is circulation of bacteria in the bloodstream, pneumonia, that is inflammation of lungs, wound infections, then sepsis neonatorum in newborn, and meningitis, inflammation of meninges in the brain. Once more, these infections are caused by proteus when it leaves intestine and in patients with low immunity. Microbiological diagnosis, treatment, and prophylaxis, or prevention of proteus infection, infection caused by proteus. Firstly, before identification, we can collect specimen from the patient. Once more, let's come back to diseases. The collection of specimen depends on diseases in patient. If it is urinary tract infection, we should collect urine. If our patient has bacteremia, we should collect blood. In pneumonia, I hope that you have understood what can we collect in pneumonia. It's sputum or pleural fluid. In wound infections, wound discharge. In sepsis neonatorum, it's also blood. In meningitis, it is cerebrospinal fluid. So let's look here. Specimen depends on infection. Urine, pus, blood, sputum, cerebrospinal fluid. That's why it's very important to know which infections are caused by proteus. Methods that are used in diagnosis of proteus are similar to Klebsiella, and remember that most bacteria and most infections are diagnosed by the same method. The first one is microscopic method that includes preparation of smear and direct microscopy with gram staining. Look here, it's smear with gram staining. You can see that bacteria are gram-negative rod-shaped bacteria. Then bacteriological method or culture method is like in Klebsiella. When you answer our questions of your teachers at class, please don't tell that bacteriological method is like in Klebsiella. It means that you must tell this method once more. The first day, the second day, the third day. Be ready that your teacher will ask you what do we do on the first day, on the second day, and on the third day of bacteriological method. Serological tests are also used. They are used for detection of antibodies. They are the same: immune electrophoresis and agglutination test. And it's also polymerase chain reaction. It's molecular biological method. What about prophylaxis or prevention and treatment? There are no any vaccines for prophylaxis of diseases caused by proteus. Immunity doesn't develop after disease. It means that patient can have disease a lot of times during the life. Which antibiotics are used for treatment? Sure, firstly, please memorize, please remember, I ask you. Especially if infection caused by Klebsiella, proteus, or Pseudomonas is the next pathogen that we discuss. In case of hospital infections, firstly, disc diffusion method is used for detection of antibiotic sensitivity of pathogen. We can use ampicillin, cephalosporin, and aminoglycosides. Cephalosporin is a big group of antibiotics that includes ceftriaxone, ceftazidime, cefuroxime. All these drugs are started from ceph. So Klebsiella and proteus have different resistance to this pathogen or to this antibiotic, so we should know. We should use the disc diffusion method for detection of the most effective antibiotic. The another pathogen is Pseudomonas aeruginosa. Pseudomonas aeruginosa. Biological properties of Pseudomonas aeruginosa. Family is Pseudomonadaceae. Genus is Pseudomonas, and species is Pseudomonas aeruginosa. Morphology of this pathogen. It's gram-negative, rod-shaped, slightly or sometimes slightly curved bacteria. They are motile, they are lophotricious. They have flagella that extend from one end of bacterial cell. They are non-capsulated and non-spore forming. How to memorize a lot of information about bacteria. Firstly, please memorize that, for example, in this lecture, all bacteria are gram-negative. All 4 pathogens that we discuss are gram-negative. Then, this semester, we discuss just non-spore forming bacteria. So this semester, all pathogens that we discuss are non-spore forming. It's how to memorize. Cultural properties. This pathogen is obligate aerobe. Please pay your attention. Klebsiella and Proteus were obligate facultative anaerobes. Pseudomonas is obligate aerobe. It needs complete the presence of oxygen for growth. It can grow on simple medium like meat-piptin agar, and there is a special medium, is Muller-Hinton medium. Bacteria produce smooth round colonies with a fluorescent greenish color. You may see it on the petri dish. McConkie agar is also can be used. Bacteria produce colorless colonies. The specific property of Pseudomonas aeruginosa is the production of bluish pigment, two pigments that are pyocyanin and pyoverodin, which gives a greenish color to the agar. These pigments gives green color to agar to pus when it's produced in infection, or to wound. Wound becomes greenish colored. Biochemical properties of Pseudomonas. It's oxidase positive. It produces oxidase and doesn't ferment lactose. It is a main property. Its main property is that specific for Pseudomonas, because in morphology, this pathogen is similar to Enterobacteriaceae, but some Enterobacteriaceae bacteria produce ferment lactose. This pathogen doesn't ferment lactose. It has O antigen that is somatic, that is present in cell wall of bacteria. It's lipopolysaccharide of cell wall, and it's motile, so it has H antigen that is present in flagella. It's protein. Pseudomonas aeruginosa is one of the most dangerous bacteria that is present that cause hospital infections. This pathogen produces great amount of different enzymes for destruction of human tissues and organs. This pathogen have a lot of virulence factors. The first one, it has pili for adherence to mucosal membranes. Then it has some strains have capsule or slime layer. Here it is written non-encapsulated. Some strains that are present that cause infection at hospital, they have capsule or slime layer that prevent phagocytosis. Then this pathogen produce elastase. This enzyme destroy elastin in skin, lungs and blood vessels. So it destroys skin tissue, lungs and blood vessels. It produces proteases that destroy antibodies. It produces two types of toxins, exotoxin and endotoxin. The first one, that is exotoxin A, causes tissue damages in pulmonary infections, in pneumonia for example, dermatonecrosis in burns, and destruction of cornea in ocular infection, of eyes. It also produces endotoxin that causes fever and shock because it's gram-negative bacteria. And this pigment that is pyocyanin that gives greenish color cause tissue damage and inflammation. Epidemiology and Pathogenesis of infections caused by Pseudomonas. The sources of infections are sick men or bacterial carriers. These infections are transmitted by contact transmission, direct contact, and airborne transmission. There is no fecal-oral. Pseudomonas aeruginosa is pathogenic in devoiding of normal defenses, in intravenous or urinary catheters, disruption of skin and mucosal membranes. This pathogen cause infections when normal defenses are destroyed. But this pathogen has a great amount of virulence factors that it causes infections very fast. Which infections are caused by Pseudomonas? These are wounds and burns, and the specific property of wounds and burns infections caused by Pseudomonas is a blue-green pus that is produced. Meningitis, it's inflammation of meninges in brain that occurs in neurosurgical procedure, in operations, for example. Then it also causes urinary tract infections that occurs because of catheters and instruments using. It causes pneumonia after tracheostomy. Maybe you know what is tracheostomy. It is a rejection of throat for injection of a special tube when patient is not able to breathe himself. So infection can be got after tracheostomy procedure. Otitis media is inflammation of middle ear. Endocarditis is inflammation of endocard in head. Eye infection occurs in injury or some surgical procedures, some operations on eye. And bacteremia, that's circulation of bacteria in the blood. Pseudomonas aeruginosa is one of the most dangerous pathogens of all surgical departments, surgical hospitals, operation halls, and burn departments when there are patients with burns. If Pseudomonas aeruginosa is detected at hospital or at any department, it must be closed. For full washing, for full cleaning, for full disinfection. Microbiological diagnosis, treatment, and prophylaxis of or specific prevention of Pseudomonas infections. Once more, before diagnosis, we should collect specimen from the patient. Which specimen may be collected? It depends on infection. It's pus, urine, blood, spinal fluid in meningitis, sputum in pneumonia, eye and ear discharge in infections there. Which methods are used? Methods are the same. The first one is microscopic. Microscopic method includes preparation of smear and gram staining and detection of its shape in gram staining. Bacteriological method is like in previous bacteria in Klebsiella and Proteus. In addition to this method, we should consider oxidase positive test and production of pyocyanin that shows, that detect, that identify this pathogen. Then serological tests that are used for detection of antibodies. This is enzyme-linked immunosorbent assay, or ELISA, the short name of this method. and molecular method, that is PCR. What about prevention? There are no licensed vaccines for clinical using. They are not used. There are no vaccines. What about treatment of infections caused by Pseudomonas? Pseudomonas is one of the most antibiotic-resistant bacteria. The World Health Organization considers this bacteria as the most resistant to great amount of antibiotics. So Pseudomonas infections must be treated with combination at least of 2 antibiotics due to its resistance. 2 antibiotics together. Which antibiotics can be used? This is tobramycin, carbapenems, cephalosporins, ceftazidime, cefoperazone. These ceftazidime and cefoperazone are also from group of cephalosporin antibiotic and aminoglycoside. Those students who will work at surgical departments, surgical or urinary departments at hospitals, you must memorize, you must remember about. Sure, any student must remember about Pseudomonas, but everyone who is going to be a surgery or a urinary doctor, you must remember about this pathogen. And the last pathogen for today is Helicobacter pylori. Helicobacter pylori, from the name of this pathogen, pylori. If you studied anatomy, I hope that you studied anatomy, you may understand that this pathogen causes infections of which organ? It's stomach, because pyloric a part is present in stomach. Family Helicobacteriaceae, genus Helicobacter, and species is Helicobacter pylori. Firstly, some words about name of genus. Helios is Helios is translated as sun from Greek language. Firstly, when this pathogen was discovered, it was in form of cyst. It was a curved, and there were flagella around bacteria, and doctors who firstly discovered this pathogen said that it was similar to sun, and they called them this bacteria Helicobacter. I'm not sure that it's like the sun, but the name was given, that's why we use it today. This bacteria is one of those bacteria who discovered some years ago. In 1983, Australian doctors, Dr. Warren and Dr. Marshall, discovered the role of this bacterium in development of gastric and duodenal ulcer. They were awarded with a Nobel Prize in Medicine for their discoveries in medicine. This pathogen was discovered earlier, but it was not known that it caused infections of stomach. What about morphology? It's small curved rods, gram-negative, motile, non-capsulated, and non-sporing. This pathogen is microaerophilic. What does it mean? It needs increased amount of oxygen, about 5%, but decrease amount of oxygen, but increase the amount of carbon dioxide, about 10%. This pathogen needs selective media for growth. It's Skyros medium. Skyros is the name of doctor who suggested to use it. Skyros medium is a blood agar in combination with selective antibiotics. Selective antibiotics are used to inhibit growth of other pathogens because a pathogen, Helicobacter pylori, grows longer than most bacteria during 3 or 6 days. Longer. During this time, other bacteria can grow if they enter from air, for example. That's why we need to inhibit these pathogens by antibiotics. We also can use chocolate agar. Chocolate agar is called chocolate not because of the presence of chocolate. Blood agar consists of red blood cells. Chocolate agar consists of lysed or destroyed red blood cells. That's why its color is brown like chocolate. This pathogen produces convex circular colonies on the surface of this medium. Biochemical properties or fermentative properties. Urease test is positive. It produces urease. And it also produces hydrogen sulfide. Antigenic structure includes O antigen that is present in cell wall. It's somatic. It is called somatic. It is lipopolysaccharide. And H antigen is present in flagella. What about O antigen? In some bacteria you can see that it is called somatic. A lot of years ago, doctors detected O antigen in bacteria, and they thought that O antigen was present in body of bacteria, inside of bacteria, in soma. Soma, it is translated as a body from Latin language. But then they understood that it was present in cell wall. But name was saved. It is called somatic. Which virulence factors are present in Helicobacter pylori? The first one is urease. Urease provides colonization of gastric mucosa and provides protection of Helicobacter pylori from gastric juice. Spiral-shaped and flagella are very important for motility within mucous layer. That's why Helicobacter pylori colonize it very fast. Then bacteria produce adhesins for binding to host cells. It produces mucinases and phospholipases that disrupt gastric mucus. It is gram-negative, so it produces endotoxin. They are microaerophils that is important for surveillance within the mucous gel in mucous membrane. It also produces a vacuolating cytotoxin that induces vacuolation in epithelial cells that results in epithelial cell damage. And Helicobacter produces acid inhibitory protein that blocks secretion of acids in stomach. A large amount of virulence factors. Epidemiology and Pathogenesis of diseases caused by Helicobacter pylori. Sources of infection are sick men and bacterial carriers. It's transmitted by fecal to oral, from stool to mouth, after toilet, for example, or oral to oral, from mouth to mouth via stomach contents when humans use the same bottles they touch with the mouth, for example, then when they use the same plates, dishes or cups, and that's why family members often carry the same strain. Because family members in one family use the same dishes. Diseases that are caused by this pathogen includes duodenal ulcer, gastric or stomach ulcer, and stomach cancer. What about pathogenesis? How disease develops in human organism? When we talk about pathogenesis, you must imagine it in your mind how pathogen move in stomach, for example, here. The first one, pathogen adhere on stomach mucous membrane. You know it's with using of adhesion proteins. After adherence, bacteria blocks the production of acid, gastric acid, because gastric acid is very harmful for bacteria by its acid inhibitory protein. Then bacteria neutralizes the gastric acidity, because there is gastric acid around the bacteria. How do bacteria neutralize gastric juice? By splitting of urea into ammonia. They produce urease that ferment urea into ammonia, and it makes pH alkaline. That is good for bacteria. Then bacteria colonize gastric mucosa. Colonize, it means they multiply. They produce mucinases, phospholipases, and vacuolating cytotoxin that damage mucosal and epithelial cells in stomach. Now imagine that a stomach of the patient doesn't have any mucosal cells. What is the main role of mucosal membrane in stomach? It protects epithelial layer from gastric juice. But now mucosal layer is destroyed by this pathogen. That's why, then look here, gastric juice contacts with stomach epithelium and contacts during long period. It causes irritation and inflammation of stomach mucosal membrane. This disease is called gastritis, inflammation of stomach mucosal membrane. Then it causes destruction of epithelium. It is gastric ulcer. When Helicobacter pylori present in stomach during a long period, this infection causes atrophic and even metaplastic changes in the stomach cells that leads to gastric cancer. In these diagrams, in this picture, you can see manifestation of duodenal ulcer in the left diagram. Duodenal, it's here. Look here in the picture, duodenum present here, and it is ulcer in this place. And gastric ulcer, it is stomach, it's present in this part of stomach. What about diagnosis, treatment, and prevention of infections caused by Helicobacter pylori? Specimen includes collection of stool, gastric biopsy specimens for histological examination, and blood is collected for detection of antibodies. Which methods are used? The first one is microscopic, the second one is bacteriological, that includes inoculation of specimen on scarce medium. Serological test is used for detection of antibodies. We can use an ELISA and polymerase chain reaction. There is specific test for detection of Helicobacter pylori. It is urease test. Urease test is based on ability of Helicobacter pylori to produce urease. Once more, urease is enzyme that split urea into ammonia. Gastric biopsy can be placed into urea containing medium into urea containing medium with color indicator. So we place it into medium that contains urea and indicator. If Helicobacter pylori is present in this biopsy, urease rapidly splits urea and resulting shift in pH. pH become alkaline and color change in the medium. We can see that it is positive result. Color of the medium is changed because of the presence of Helicobacter pylori that produce urease. This test is negative, it means the pathogen is absent. Vaccine is not available for prevention of diseases caused by this pathogen, caused by Helicobacter. What about treatment? Triple therapy has prompt response containing combination of following drugs. 3 drugs that are used for therapy of gastric ulcer or duodenal ulcer. It's metronidazole, the first one, it's antimicrobial drug. Then bismuth subsalicylate that decrease acidity of gastric juice. And amoxicillin is antibiotic. It is administered up to 14 days. So Helicobacter pylori also becomes resistant to antibiotics, so we use here 2 drugs. Metronidazole is antimicrobial drug and amoxicillin is antibiotic. Bismuth subsalicylate is not antibiotic. It is used for to decrease acidity of gastric juice, because the main role in development of gastric ulcer in gastric juice when it contacts with epithelial cells. Now that's all. Thanks for your attention. I remind you that next week you will start to have colloquium. Thanks for your attention. If you have any questions, you may ask these questions at practical classes. Take care of you. Goodbye.